morning, ladies and gentlemen. Welcome to the beautiful city of Los Angeles. I know we have people coming from all around the world. This is really has been an exciting, conference, from my perspective, and there has never been an exciting time in stem cell research. Every years, we're coming up with very significant and major, discoveries. I'm not going to go through a lot of them, just give one example in the context of my interest in type 1 diabetes. Last year, there were two papers came out demonstrating that human embryonic stem cells or induced pluripotent stem cells can effectively be converted into beta cells making insulin. And beta cell transplantation, and clearly for the treatment of type 1 diabetes particularly, is an important therapy, but unfortunately, the lack of sufficient, donors has been one of the major impediments. So I'm very much excited about these new developments, and this is just indicate how important this field is. One nice thing about this particular conference, we have speakers coming from all over the world with diverse research interests, but stem cells being the major theme. Every single time I come to this conference, I always learn something new and go home, with much more expanded knowledge of stem cells, which certainly applies to my own research. And basically, I want to just take a few minutes and to thank the organizing committee. We have Dr. Paul Davis, George Weber, and Diana Anderson, and particularly on the part of Omics Group, Jesse Casper. He's really done a great job communicating with everybody and putting this program together. So we are going to start with this morning session, and then we have basically tomorrow and Wednesday we're going to conclude the conference. If we have a special session for graduate students, postdocs, and junior scientists with Dr. Paul Davis. On Wednesday, we'll be talking about the opportunities, in biomedical fields for young, investigators. If anybody's interested, we look forward, to seeing you there. So with that, I'm going to go ahead and basically maybe I'll show you, let me do this. Let me start maybe my talk, and then I will introduce everybody and a member of the panel. We have, okay, so I guess all right, I'm sorry, let me do this, okay? So we don't have everybody giving a talk, so I was asked whether I could, introduce the honorable guest. We have here, I'm gonna start with Paul Davis, and Paul, you have a talk this morning, right?
Yes, I do.
Okay, so let me skip you then. I apologize. When during your talk I'll introduce you. I'm gonna start with in that case Dr. Anderson. I'm trying to find your info. A little bit disorganized. So we have Dr. Diana Anderson. She's coming to the conference from University of Bradford, United Kingdom. She completed her PhD in the Faculty of Medicine, University of Manchester, UK. She holds the established chair in biomedical science at the University of Bradford. She has published tremendous number of papers, 450, and many books. She served on 10 editorial boards, has supervised over 30 PhD students. She's the editor-in-chief of the book series Issues in Toxicology. And she has hosted and participated in 56 meetings for the World Health Organization and chemical safety. She has been a vice president of the Institute of Biology. Welcome to Philadelphia. And we have Dr. George Weber from University of Cincinnati. George has made contributions to metastasis research by discovering the interaction between the molecules osteoplantin and CD44, and by defining the physiological role of metastasis genes as stress response genes. while he continues to address fundamental questions, he's researching new venues for diagnosis therapy for cancer and progression. Welcome, George. I think, well, with this, I'd like to go ahead and maybe start, my talk here. Haval Shirwan, I'm from the University of Louisville. I'm a faculty in the Department of Microbiology and Immunology School of Medicine. And today I'll talk about our research on, let me see whether I can get this right. You don't have a mouse, do you? Do you have a mouse or a laser pointer? You don't have a laser pointer? Yeah, please. So this is basically the targeting of my talk, specific targeting of pathogenic T cell for induction of tolerance to pancreatic islets for the treatment of type 1 diabetes. So I will present this talk, but in the context of this talk, I'll present the technology where we apply, to stem cell research. And today I'm not going to talk about the application, but I'll just give you a few examples how we can utilize this particular technology for effective bone marrow transplantation or transplantation of foreign cells. For example, as I mentioned, the beta cells derived from human embryonic stem cells for the treatment of type 1 diabetes. So type 1 diabetes is an autoimmune disease. As you know, T cells are the major culprit of type 1 diabetes. They recognize beta cell-driven antigens, and their response to beta cells destroy beta cells. The end result is diabetes. There are genetic as well as environmental, risk factors. I'm not going to go into those, but it's a pretty devastating disease, and the major complication simply is kidney failure, blindness, nerve damage, heart attack, and stroke, amputations. It affects 3 million people in the United States. And most importantly, the incidence of type 1 diabetes actually is rising, and we don't know why that is the case, but certainly it's becoming a major health problem. There are no preventive, treatments, even though the diagnostic technologies allows us to identify people with the potential of developing type 1 diabetes, but really we don't have a way to prevent the development of the disease. So that's our problem. The treatment presently, pancreas transplantation, this is particularly is done, pancreas and kidney combination usually is done, but that's very invasive, clearly not the major surgery. Usually pancreas by itself is hardly done. Islet transplantation, pancreatic islet transplantation, perceived to be a means by which we can treat type 1 diabetes. The problem there is there are a couple of problems. Number one, we have to use immunosuppression to control rejection, and number two, simply the paucity of the available donors. As a result, there are attempts to use porcine as potential source of pancreatic islets, and in that case, again, the issue is really the immunological barrier for a xenotransplantation is extremely high. The immunosuppression effectively being used in allo setting doesn't necessarily control rejection of xeno islets. And that's why, as I pointed out, this new developments that we can drive beta cells, human beta cells, from embryonic stem cells is becoming so exciting. So that remains to be seen. Even in that context, clearly we need to still deal with allo rejection. If we drive from the same individual, autoimmunity is still going to be a problem at the end of the day. We have to define or come up with an approaches to effectively regulate immune responses. Presently, clearly the type 1 diabetes is being managed by exogenous insulin treatment, and that has its own major problems, unfortunately, and the quality of life is clearly not that great. So as I indicated earlier, and this is actually not only the major problem for type 1 diabetes, but current understanding is that every autoimmune disease is suffering from an imbalance between the so-called the protective T regulatory cells and the destructive T effector cells. So under normal conditions, there is a balance here. T regulatory cells are self-reactive. They keep in check the pathogenic T effector cells, and we live a healthy life. When that balance, for whatever reason, is perturbed, and the end result, we have your T effector cells amplifying and basically destroying beta cells making insulin. And this is, of course, is true in the case of graft rejection. If you take foreign beta cells put into an individual, if you cross a minor or major histocompatibility barrier, come down with this reaction. T effector cells eliminate the transplanted graft. So the question is that can we reverse this balance? I mean, that is, if we could effectively do this, this can apply to multiple sclerosis, can apply to lupus, arthritis, and etc. So this is our goal, and this is my favorite cartoon demonstrating the interaction between a T cells and an antigen-presenting cell, and these are the key cell surface receptor and ligand interactions. So immune system make a decision once it recognizes the antigen in context of T cell receptor, and then you get a co-stimulatory signal, and then signal 3 is in form of cytokines, and this is basically what happens. You amplify T cells and then make effector cells. The effector cells deal with the infection. So that's how the immune system functions. And once the immune system deals, and this is the major purpose of the immune system, actually to protect us against infections, right? And once the immune system deals with infections, and what happens is that sorry about this. Then we have a lot of activated T cells in our body. They have to be eliminated, and the way the immune system deals with these cells, they basically apoptose those cells. So they get rid of a lot of these apoptotic cells, these activated cells, and this is called activation-induced cell death. The way this happens is that when naive T cell, they don't express fast or fast ligand. There's a death receptor and ligand. Once they get activated, they express both fast and fast ligand. But the T cells, they are resistant to apoptosis. There are anti-apoptotic genes that regulate the apoptosis. And then once the level of the immune system deals with infection, the levels of anti-apoptotic genes basically goes down, and these T cells are being eliminated. There are a few T cells left. They go on establish immune memory. That's how this immune system function, and fast ligand plays a major role, and that is the reason. And another important evidence demonstrating the importance of fast ligand mediated pathway in immune homeostasis is that autoimmune lymphoproliferative syndrome in humans is associated with mutation with FAST. Deficiency in fast ligand in rodents results in excessive autoimmunity. Indeed, you could correct autoimmunity transgenically, expressing fast or fast ligand. This evidence indicates that there is no compensatory mechanism for fast ligand, and indicate its importance in immune regulation. And that's why we basically focus on this pathway. I guess it's going to become pretty annoying. I don't know how to turn this off. Okay. So, okay, thank you. I'm not going to go through this because I want to discuss some of the data, the way it works. There are two extrinsic pathway. This is receptor-mediated that pathway, and there's intrinsic mitochondrial pathway. And when fast ligand bind to fast, and fast ligand is a cell surface molecules from trimers, and then recruit basically some other molecules that ends up in the activation of caspase 8. Caspase 8 activates caspase 3, and caspase 3 basically causes cell death. And so, this is going to become pretty annoying here. I'm going to say minor issue. Go ahead and. Yeah, I think that's the best way. Just to. How do you disconnect this? Sorry. Airplane mode. Okay. Thank you. See, he knows what he's talking about, right? Okay. All right. So we're getting that. So I'm
Ilyas,
test case, I guess. So what we wanted to do, we wanted to basically simulate this
you already have your presentation.
physiological activation in the cell death, and the way to do that, we thought we could. We're gonna have a lot of issues here. I guess we're gonna. Okay, let me just. Yeah, we are going to start in just a couple of minutes. Naveen-equivalent solution for this because it's going to be very annoying. So what we thought we could do, we could simply take the G4 fast ligand and then use gene therapy to express it in donor cells or tissues. And then when we transplant the donor cell and tissue, this could be your stem cells or hematopoietic stem cells or beta cells. When immune response recognize the foreign tissue and response, that T cells will get activated, express fast on the surface, fast will engage fast ligand, and induce apoptosis that specifically will purge out those pathogenic T cells. So that was the concept we entertained. This is quite some time back. We managed to get the fast ligand gene and try to use a lot of different vectors to express it effectively in cells or tissues, and this is kind of keeping clinic in mind because we wanted to, once we identify, for example, a recipient and donor, we should be able to get the donor cells like peripheral blood lymphocytes or stem cells, transfect them or transduce them with the gene, and then within a few days, give it to the patient, purge out alloreactive cells. So it turned out to be almost an impossible task, to do that, and we finally gave up, and then we focused on this particular concept. It's called PROTEX. What this concept is that it allows us to generate recombinant proteins with potent immunomodulatory activities, and then we can use these proteins either as soluble biologic, or we can actually stick it on the cell surface and to accomplish immunomodulation, as I indicated earlier, because the immune decisions are end result of effective cell surface ligand and receptor interaction, and this interaction basically takes place within 10-15 minutes by some estimate. The immune system makes that decision and basically chart that course. So we thought we don't really need to use gene therapy to do this. If we manage to keep this ligand on the cell surface for an extended period of time in order of, let's say, 10-12 hours, we should be able to accomplish the same goal of using gene therapy to express this protein. Any questions? All right, if not.
Question here.
This one. I'm sorry, go ahead.
So, very nice job. So, I'm wondering, the fast ligand can kill a lot of cells. So, when you engineer your cells, so you kill the, effector cells, but why it doesn't kill the host, cells? The cells, you inject into the,
So, in this context, we are not injecting the cell. These are pancreatic islets.
Yeah.
So when
So eventually you inject into the, the pancreas of the animal, right? You did the experiment, right? So,
Right.
I'm wondering why it doesn't kill the, the cells around, in the pancreas, the host of the pancreas.
well, that's a good question. This is something, whether, if I understood the question correctly, the beta cells, pancreatic beta cells, there were some reports they may be sensitive to fast ligand mediated killing. There were some papers published way back, and there were a bunch of other papers demonstrating that's actually not the case. Under inflammatory conditions, those cells, they may express a fast on their surface, but beta cells they consider to be a type 2 cells. I'm not going to go into that definition. What that means is that those cells, basically, they use mitochondrial pathway for apoptosis, not necessarily death-mediated pathway. So we don't really see it, and I mean, this is certainly one of the issues. I think this is one of the major problems why the academia as well as industry gave up on this pathway because of this potential toxicity. But without construct, maybe it is the attribute of the construct, we don't see it. And another thing, what people have shown that that JO2 antibody that's used by Nagata and co-workers, it appears really that's the unique attributes of that antibody because they have the Fc portion, they bind the Fc receptors, and they do a bunch of other things. So we don't see it. I mean, that's the data. We're not only in rodents, we've gone into non-human primates, and we don't see it.
Do islet cells make PD-L1?
Good question. Actually, they do. If you look at the work by Mosaic and others at Harvard, they've demonstrated that when you interfere with PD or PD-L1 pathway in NOD, you induce accelerated diabetes. And apparently PD-L1 has been really important pathway. The T regulatory cells, they express PD-1, and probably PD-L1 too, but for sure as an important regulatory pathway.